Hi. I'm Peter McColgan. I'm an academic clinical lecturer in UCL and a visiting scientist in the Max Planck Institute for Human and Cognitive Brain Sciences. Today I'm going to talk about quantitative 7T MRI, cytoarchitectonics, connectomics, and regional gene expression. So I just want to give an overview of the project first. Our aim was to try and understand what we can measure with 7T at 500 microns using multiparametric maps, looking across the depth of the cortex. So to try and do this, we looked at the relationship between our MRI measures across cortical depths and von Economou cell count across cortical layers. We looked at the cell staining intensity across cortical depths in big brain. And we also looked at lamina-specific genes from the Allen Human Brain Atlas. Then finally, we wanted to try to connect the depth-related contrast at 7T with specific white matter connections based on anatomical priors. So I just want to give a brief overview about the layers of the cerebral cortex first. So the isocortex has 6 different layers. Layer 3 has pyramidal cells that form other white matter connections with other cortical regions, so these form cortical-cortical white matter connections. Layers 5 and 6 have pyramidal cells that form white matter connections with the deeper structures, particularly the striatum and the brainstem. The reason why this is important for me is I'm a Huntington's disease neurologist. So Huntington's disease is a neurodegenerative disease that affects the striatum, but work I've done previously shows that loss of white matter connections occur even before people get symptoms between the striatum and the cortex. Based on what we know from the connections and the anatomy of cortical layers, this leads to the hypothesis that there is dysfunction and death of cells in the deeper layers, followed by dysfunction and death of cells in the superficial layers, and then loss of interhemispheric white matter connections, again, which I've demonstrated previously. But to try and, before we apply this to a disease, we have to understand what we can measure and what are our limitations. So we collected data in 10 healthy controls, 500 micron resolution, and I'll focus here on R1 and R2 star. In the same subjects, we also collected multi-cell 3T diffusion MRI data. We used the Scholten's MRI von Economou atlas to look at von Economou. We used Desikan to look at big brain, and we used the Glasser atlas to look at the lamina-specific genes. We used NIRES to create equivolume cortical layers and FreeSurfer to do the surface-based registrations. So this is an example of what we can do with NIRES. So this is M1, and we define the pale surface and the gray matter, white matter surface, and then we can create equivolume layers across it. So the different colors here are the different layers. The first thing we wanted to do with the 500 micron MPMs was to show that we can reproduce known myelination patterns. So R1 is sensitive to myelin, R2 star is sensitive to myelin and iron. And we can see here that both with R2 star and R1, the highest values here are seen in the motor cortex, the auditory cortex, and V1, which are in red. And we take this one step further by looking at the visual layers across the depth of the cortex. So the x-axis here is cortical depth going from pale surface to gray matter, white matter boundary, and the y-axis is R, which is a measure of myelin. And we can see that, as one would expect, myelin increases as we go deeper down the cortical layers. And again, V1 is the most myelinated of these regions. So I just want to try and explain the next part of the analysis in terms of what we did. So we had 10 brains. We averaged across them, and we had 8 layers looking at R1 and R2 star. To look at von Economou cell count, we used the von Economou atlas. So this is 8 layers times 43 ROIs. For big brain, we had Desikan, so 8 layers times 33 ROIs. And for transcriptomics, we use Glasser, so 8 times 180 ROIs. And so these analyses, the correlations are performed across all the ROIs in the cortex. So just showing what we did with von Economou. So I'm just showing this as an example to try and understand the next plots. So on the y-axis, we have R2 star at 5th cortical depth. And on the x-axis, we have von Economou cell count in layer 2. You can see here that these are analogous to the auditory cortex and the visual and the motor cortex, have high R2 star and have high cell count as well. And so if we do this for all the cortical depths in the MRI and all the layers in von Economo, we can create this type of image. So the brighter colors here are the higher correlations. And so when we do this for R2 star and von Economo cell count, the red boxes here are bond forming corrected significance. We see really high correlations of 0.7 for layers 2, 3, 4, and 6. To try and understand this a bit more, we look at the cross correlations just within the von Economo cell count data itself, and we see that layers 2, 3, 4, and 6 are highly correlated. So this explains possibly why we're not seeing a one-to-one correlation across layers and depth. Again, for big brain, this time we look at big brain intensity profiles. I took 1,000 profiles and averaged them across 6 arbitrary divisions. And again, we look across depths for R2 star and the MRI data. We see high correlations with intensity profiles 2, 3, 4, and 5. And again, it's even higher this time. So correlations of up to 0.8 with our R2 star data. And again, try to tease this apart a little bit more. We look at cross correlations and we see that the intensity profiles 2, 3, 4, and 4, 5, 6 are highly correlated. The next step was to look at regional gene expression. So I first did an ROI analysis. So each dot here is an ROI from the Glasser Atlas. And we have the average R2 star. And then we have lamina-specific genes for layers 1 to 3, layer 4, and layer 5 to 6. We do a PCA on these genes and then take the first component. So we have gene expression for each ROI. And you see that there's high correlation with layers 1 to 3 and layer 4, but not with layers 5 and 6. And when we do this across depths, we see a similar pattern. However, the correlations get higher as we go down the depth of the cortex, getting maximum at depth 5 and 6. So I've shown that R2 star is highly correlated with von Economo and is highly correlated with big brain. And I also want to mention that R1 did not show bond Ferroni corrected significant correlations, but this is possibly related to noise and is something that we're looking into. For the connectomics, I split white matter connections up into corticostriatal, corticothalamic, corticocortical, interhemispheric, and intrahemispheric. And again, I looked at correlations across the depth of the cortex. And we don't see any bond Ferroni corrected significant here for these white matter connections. And this is when we weight by streamlines. However, if we weight the white matter connections by R2 star and look at, so R2 star in the white matter and look at R2 star across the cortical depth, we begin to see correlations with corticostriatal and intrahemispheric connections, up to 0.7, so very highly correlated. Again, if we take R1 across cortical depths and weight the connectome with R1 and the white matter, we see really high correlations for corticostriatal connections. And the correlations are the highest at the deeper layers, which fits what we know about the anatomy. So I just want to summarize, R1 and R2 star at 7T shows no myelination patterns. R2 star has strong correlations with lamina-specific cell count and lamina-specific genes. R2 star and R1 has strong correlations with intrahemispheric and corticostriatal weighted white matter connections. And I think this suggests that if we apply this type of framework to disease, it's neurodegenerative disease, we can begin to track changes in white matter and gray matter using these approaches and to get some lamina specificity. I just want to thank everyone who's been involved in the work, particularly Professor Nicholas Weisskopf, Sarah Tabrizi and Geraint Rees, and everyone in the Max Planck and UCL and the Dutch Connectome Lab as well.